Hello students, welcome to the new lecture, and in this lecture we are going to discuss about the new topic, okay. So that is blood grouping. Okay, by the way, good afternoon everyone, good afternoon. Okay, so welcome to the new lecture, and this is about the blood grouping. Okay, so what is the meant by blood? In the last chapter, in the previous chapters also, not the chapter, just lectures. In the previous lectures we have discussed the blood is a fluid connective tissue. Okay, what is the blood? Blood is fluid connective tissue. Okay, fluid. Okay, so fluid connective tissue. So why we are calling this blood as a fluid connective tissue? Why? Because the blood looks like fluid, and it is going to have the blood cells. Okay, so and also it is going to connect different parts of the body. Okay, so it is the reason. This is the reason it is a fluid connective tissue. Okay. So here a important person, he is known as Landsteiner. I think we have discussed the blood grouping in the last class. Okay. So what he said, he discovered that blood groups, human blood groups has a four groups, and also he divided into four groups. They are A, B, AB and O. So these are the four blood groups in human beings. Okay, and also we have discussed about universal donors, means the person who is having the universal recipients. Universal recipient means the person who is having the AB blood group. The person who is having the AB blood group can receive blood from any group. Okay, from the person who is having any blood group. So this is the reason they have the capability of accepting any blood group from any person. So they are known as universal recipients. While the persons who are able to donate blood to any person, they are known as universal donors. Okay. And by the way, if you have any doubts, okay, if you have any doubts or questions, so feel free to post in the live chat section so that we can discuss those doubts and questions. Okay. So by the way, good afternoon to everyone, and now we will move to the new topic, that is lab activity. In this chapter, in this class, the new topic is lab activity, and along with this lab activity, we are going to discuss about muscles. Okay, different types of muscles. Okay. So lab activity. So our aim is identification of the blood groups. So what are the blood groups? Just now we have discussed. There are four types: A, B, AB and O. These are the four types of blood groups. Means you have any one type of blood group in your body, any type, either A or B or AB or O, any one type of blood group is present in your body. So how do you find out your blood group? That is the lab activity over here. Okay. So first one is the aim. Our aim is to identify the blood group. So apparatus is a blood group identification kit. Kit is nothing but it is some factories or some companies, they are going to sell the chemical substance. So in the form of kit, with the help of that kit, we are able to identify the blood group. Okay. So next, glass slide, wax pencil, disposable needle, and so on. So these are required. So this is a table which is describing about what are the different types of materials present in the kit. Okay. So the kit contains different components like anti-sera A, okay, anti-sera A, anti-sera B, anti-RHD sera, okay, and along with it, you may require porcelain white plate, wax pencil, needle 24G. 24G means 24 gauge, okay. So 24 gauge is nothing but how much length the needle is, okay. So that is about 24 gauge. So next is instruction manual. These are the things that is present in the kit component. So here these are bottles, anti-sera A, anti-sera B and anti-sera D. These are the glass bottles. Their content is about 5 ml. So with the help of this kit, you can perform 100 tests. It means you can check the blood group of 100 people. Okay. So this is about it. Okay. So the procedure, how do you perform the lab activity and how do you identify the blood group of a person? So for this, you have to take a clean and dry porcelain plate. So this is a clean and dry porcelain plate, and also we have to draw 3 circles. Okay, so we have to draw 3 circles. So as shown in this diagram, you can see a small porcelain plate, or instead of porcelain plate, you can take a slide. Okay, and with the help of wax pencil, we have to draw 3 circles and name each circle as A, B, and also RH. So this thing should be drawn and also written on the porcelain plate. So now we have to put a drop of corresponding anti-sera. So what is anti-sera? Here you can see in the kit we have some bottles, no, 3 bottles there. So these 3 bottles are the antisera. Okay. So here antisera A, antisera B, and also antisera D, or you can call it as RHD. So in the first circle we have to add antisera A. Here we have to add antisera A, and in the second circle we have to add antisera B, and in the third circle we have to add antisera RHD. Okay. So it means to the corner, to the edge of the circle we have to add one drop. Okay. So one drop from the each bottle we have to add. Okay. So next, choose any finger. So choose any finger and also cleaned with alcohol, cotton ball containing alcohol. So why we are using the alcohol with alcohol? So alcohol, it is going to kill the microorganisms. Okay, it is going to kill microorganisms. Yeah, on every surface, whether it may be your finger or your hand or table, chair, or whatever it may be, everywhere the microorganisms are there. So while puncturing the finger with the help of needle, some microorganisms may enter into the body. Okay, some microorganisms may enter into the body. So we have to prevent it. We have to prevent the entry of microorganisms into the body. So as a result, what we have to do? We have to kill the microorganism. We have to kill the microorganism by using alcohol, cotton ball containing alcohol. So if you rub the cotton ball containing alcohol, alcohol is going to kill the microorganisms. Then it is sterilized. Your finger is sterilized and it is ready to prick. Okay, prick is nothing but pierce with the help of needle. So pierce into a nail. Okay, apply small pressure with the help of needle on the finger. So that is nothing but pricking. So if you prick it, a small if you apply some amount of pressure on the finger, that is nothing but pricking. In that case, a small little bit amount of blood will come out of the finger. Okay. So because of the needle pricking, so small amount of the blood will come out. So then press the finger while the blood is coming. Press the finger and also put a one drop on each circle. So here one drop, here one drop, and also here one drop. So 3 drops are placed in the circles. So previously we have added antisera A, antisera B, and also RHD antisera. Now what we have added? We have added the blood also. Okay, in each circle one drop of blood. Okay. Now what we have to do? We have to mix them. Okay, with the help of toothpick. Take 3 toothpicks. Okay, 1, 2, 3, and also we take the 3 toothpicks and take first toothpick and also mix the first circle. Okay, mix the contents which is present in the first circle. It means we have to mix antisera A drop and also blood drop. And also take second toothpick and also mix the antisera B and also blood which is present in the second circle. Third toothpick and mix the contents in the third circle. So make sure you should not use the same toothpick to mix other circles. So in that condition the cross contamination will take place and results will not be shown. Okay, so this is the procedure. So we have to wait for some time. Like around 5 to 6 minutes. So what will happen? The agglutination will occur. What is meant by agglutination? Here the RBC, they are going to come together and they are going to form a clump. Okay, they are going to form a clump. It means they are going to form a group of cells. So this group of cells, this group of RBC cells that will come together, that is known as agglutination. So here the reaction is taking place between the antisera and also blood group. As a result, at the end of the reaction, there will be a identification of the blood. Okay, identification of the blood group. So here the RBC are suspended. RBC are suspended means RBC cells are suspended in the solution containing antisera and also blood. Okay. So this RH is sometimes slower to agglutinate. So it may take generally it will take around 5 to 6 minutes. Sometimes it may take around 10 minutes also. So have a patient and also wait for a little bit more time. So next, RH factor of the blood group can be identified based on the determination of RHD antiserum. Okay. So based upon RHD, here you can see RHD. Based upon the RHD only you can identify RH factor. Okay. So for example, this table is going to explain better. So here what is happening here? The blood group of 4 persons has been taken. Okay, first person, second, third, and fourth. So what happened in the first slide? Okay, in the first slide, our first porcelain plate. In the first circle are anti-A the agglutination is taken place, and anti-B there is no agglutination. So and here in the type, so what is happening here? The type is A. Why? Because and here you can see the agglutination is seen in the anti-A, so agglutination is seen in the anti-A circle, so the blood group is A. Okay, blood group is A. So for example, in the third circle, okay, let us consider the three circles. Okay, so the agglutination is seen in this. Okay, no agglutination, and agglutination is seen in this. So what will be the blood group? Agglutination is seen in the anti-A, no? So A blood group. So agglutination is seen in the RHD also. So if agglutination is seen, the RH will be positive. So A positive. Okay. Let us consider. So second plate. So in the second plate, three circles are there. So agglutination is not seen. No agglutination in the second one. Okay. In the first circle, and agglutination is seen in the second circle, and agglutination is seen in the RH also. So what will be the blood group? The blood group will be B. Okay, B, and agglutination is seen in the RHD, no? So it will be positive. Okay, B positive. So let us take another test. Okay, so in another test, the three circles again. Okay, the three circles again. So here agglutination is seen here, agglutination is seen here, and also agglutination is not seen here. So what will be the blood group? The blood group will be A and B. Okay, agglutination is seen in antisera A and antisera B, no? So agglutination will be seen in the antisera A and antisera B. So blood group is AB. So agglutination is not seen in the RHD. So it is negative. So AB negative blood group. So next one will be, so another circle. Okay, another test. Okay, another test. So agglutination is not seen here. Agglutination is not seen here. What will be the blood group? The blood group will be O. Okay. So if the blood group is, the agglutination is seen in the RHD over here, it will be positive. If the agglutination is not seen in the RHD, in that condition, it will be negative. Okay. So this is about RH antigen identification along with blood group identification. Okay. So I hope this topic is helpful and also you have understood. If you have any doubts or questions, feel free to post your doubt or question in the comment section, or that is nothing but in the live chat section, so that we can discuss about your doubt or question. So next one will be the muscular tissue. So what is muscular tissue? Muscular tissue is nothing but these are a type of muscle cells that is present in the body for different functions. What are the different functions? For locomotion, okay, for the locomotion, okay, so for the digestion and also excretion and so on. These are the different biological functions. In all these biological functions, these muscular tissue is helpful. So muscle tissue consists of elongated cells. Elongated cells means these cells are long, okay, these cells are long, and also they are called muscle fibers. So these the elongated cells is present in the muscle tissue. Those cells are known as muscle fibers. Muscle fibers are the cells which is present in the muscle tissue. Okay. So muscle fibers containing the special protein called contractile protein. We are calling it as a contractile. Okay. We are calling it as a contractile. Why we are calling it as a contractile protein? Why? Because these contractile proteins are helpful in the contraction and relaxation of the muscles. Okay. So due to this reason, due to the contraction, relaxation of the contractile proteins, we are calling these proteins as contractile proteins. So these contractile proteins are going to help in the movement of the organism. For example, if you want to move from your home to shop, you are using your muscle tissue. Muscle tissue contains muscle fiber cells, and these fiber cells contains contractile proteins that is helpful in the movement, okay, by contraction and relaxation. Okay. So here what is happening here in the winter season? There is very cold outside, so your body has to maintain some temperature. So in that condition, these muscle fibers, they are going to contract and relax. So contraction and relaxation of the muscles is going to help in the production of heat, okay, the production of heat. So contraction, relaxation of the muscle tissue is going to help in the production of heat. So this heat is maintained in the body so that your body functions well, okay. So the muscles are responsible for the movement in different organs. So those organs are legs. So those organs are legs, hands, and so on. So if you want to move your leg from one position to another position, you have to use the muscle cells, okay. Muscle cells, there is nothing but muscle fibers. So here the internal organs also going to have internal organs like heart, kidney, and so on. So these organs also going to have some amount of muscle tissue, okay. So and also blood vessels is also going to have muscle tissue. What is meant by blood vessel? In your 10th class, in the next academic year, we are going to discuss about types of blood vessels. So this is a blood vessel, okay. What is blood vessel? A blood vessel is nothing but a tube that helps in the flow of blood through it, okay. So that is known as a blood vessel. So here the blood vessels also made up of muscles. So what is the function of the blood vessels over here? What is the function of the muscles in the blood vessels? These muscles are helping in the increasing and decreasing the diameter. So let us consider this is the diameter of the blood vessel. So if it is contracting, the muscle cells contract, so the diameter is going to decrease. So diameter is going to decrease. How can you see the diameter? Here is a larger diameter, okay, here is a smaller diameter. Means the diameter can be increased or decreased because of the muscle cells which is present in the blood vessels, okay. So next is heart is made up of muscle cells, okay. So the cells of heart or the muscle cells of the heart is known as cardiac, okay, cardiac muscle cells or cardiac cells. So these are nothing but cells of the heart, okay. So here these muscles which is present in the heart cells is responsible for the functioning of the heart. So we know that the functioning of the heart is to pump blood, okay, to pump blood. So it is a main function of the heart. So it means the muscle cells or the cardiac muscles are helpful in the transportation or pumping of blood, okay. So here the muscles are of three types, okay. Based upon the structure, location, and function, the muscles are of three types, okay. The three types of muscles are striated muscles, non-striated muscles, and also cardiac muscles. So based upon their structure, function, and so on, and also location, we are categorizing muscle cells into striated muscles, non-striated muscles, and also cardiac muscles, okay. So first one will be the striated muscles. Striated muscles means they have some striations, okay. Let us consider this is a muscle cell, okay. These muscle cells will have the striations, okay, small some kind of structures or some kind of lines on them. So these are known as striations. So these tissue are long and also cylindrical and also branched. Here you can see these cells are branched in this way and so on. In this diagram you can see the striated muscles. In this diagram you can see clearly. So these muscle cells have the cross striations, and these striations are going to help in the identification of the type of muscles, okay. So these tissue are long and cylindrical. Cylindrical means like cylinder, like round shaped, and also branched and also multinucleated cytoplasm. So multinucleated, okay. So multinucleated. So what is meant by multinucleated? So multi means many. Multi means many. Nucleated means nucleus, okay. Nucleus. It means these muscle cells are going to have many cells in the cytoplasm, okay. If you have seen no cytoplasm, what is meant by cytoplasm? This is a cell. Let us consider this is a cell. In this cell, the nucleus are many nucleus are there, and these nucleus are present in where? In the cytoplasm, these nucleus are present, okay. The cells are voluntary muscles, help in the locomotion, okay, help in the muscles, okay. So these cells are voluntary muscles. So what is meant by voluntary muscles? Voluntary, it is nothing but, okay, it can be operated on your wish, okay. If you want to move your limb, okay, so it is going to move, it will be moved, okay. It means based upon your control only they are operated. So voluntary means under your control. So these muscles are under your control. So example, inner limbs. In your limbs, So these muscles are present, these muscles are voluntary. When you want to operate it, they will get operated. Okay. So these muscle cells attach to bones, means any muscle cells that attach to the bones, and most of the muscle cells in our body is attached to the bones. So they are known as skeletal muscles. Skeletal muscles means what? The muscles which are attached to the bones in the body, that is known as skeletal muscles. Okay. So they are going to help in the movement of the body. Okay. And by the way, if you have any doubts or questions, let me know in the comment section so that we can discuss. Okay. So these points are important. So all these points that we are discussing is important. It may come in the examination. So write your notes at the same time. After completion of the lecture, again open it and also write the notes. It will be useful for you, and so that you will get some time to read and so on. So next is the non-striated muscles. Non-striated means these muscles will not have striations. It means no lines are present in them. Lines are not at all present in them. And also these muscles have the long pointed ends. So here you can see how they are pointed, means they are like some kind of a sharp point like structure. They are pointed over here. So these are uninucleated cytoplasm. It means in this cytoplasm you can see only one nucleus. So uni means one, nucleated means nucleus. They are going to have only one nucleus in the cytoplasm. So these are non-striated muscles. So these cells are present in the alimentary canal. So what is meant by alimentary canal? So alimentary canal means a long tube that is starting with mouth. Okay, starting with mouth and also ending with anus. So this is known as alimentary canal. Alimentary canal means a long tube that starts with mouth and also ends with anus. That is known as alimentary canal. So they are voluntary muscles. Okay, they are also voluntary muscles. What are the voluntary muscles? So the voluntary muscles are non-striated muscles and also non-striated muscles. So striated and non-striated muscles are voluntary muscles. Means you can operate them. Okay. So example, smooth muscles. So and controls the movements in various organs. Okay. Example, uterus, iris of eye, bronchi of lungs, etc. Okay. So these are non-striated muscles. So there is activity 5. In this activity, you have to collect 3 types of muscle slides. Okay. That is also permanent slide. So what is meant by permanent slide? Permanent slide is nothing but a glass plate, okay, small glass. So over this some tissue is placed, and over the tissue some glass cover, okay, that is nothing but cover slip is placed, and gum is attached over there. Okay, some kind of glass coating gum will be there and it will be attached. So now this slide will be permanent slide. So this slide will be permanent slide, and you can use this slide to observe anytime. So these permanent slides are made in some kind of biological companies. So they will sell it to the schools or colleges. So if you have such kind of permanent slides in the school or in the school laboratory, you can take it out and observe. So this will be about permanent slide. Okay. So non-striated muscles, cardiac. What are the muscle cells you can see? What are the permanent slides of the muscle cells you can see? So one is a striated, another is a non-striated, and also another is the cardiac muscles. Okay, these 3 types of muscles can be collected and also observed under the microscope. Okay, from the slide box. The slide box is nothing but a box containing different types of the slides. Okay, so then observe these slides under the microscope and write your findings in the following table. So there is a table which is given in the textbook, and write what you have observed. What you have observed in the cardiac muscles, and what you have observed in the striated muscles, and what you have observed in the non-striated muscles. So you have to write all these observations in your in your table, which is given in the diagram. Okay, in the textbook. Okay. So next is the cardiac muscle. So what is meant by cardiac? So the term cardiac, okay, is nothing but heart. Okay. So these cardiac muscles refers to the heart. So these muscles are present in the heart. So these cells have the striated structures. Here also you can see striated structures or lines are present in them, and they are branched. Okay, they are branched. They are long and also they are branched. They have intercalary disc. Means they are going to have some kind of disc-like structure in them, that is known as the intercalary disc, and they have long and also branched, joined at each other. It means one cell is attached to another cell. So these cells are present in the heart, and also responsible for pumping of the blood. Okay, so this is about cardiac muscles, nothing to discuss more about it. And here you can see the nervous tissue. So what is nervous tissue? Nervous tissue or neurons both are same. Okay, so these nervous tissue are a specialized cells that are involved in the transmission of the stimulus. So what is meant by stimulus? Okay, what is meant by stimulus? For example, if you are walking on the road, so suddenly there is huge horn noise came. Okay, so the sound, okay, that sound created in the environment is known as a stimulus. Okay, so now what you will do? You will respond to the stimulus. Okay, you will respond. How you will respond? Suddenly you will move out of the road. Okay, it means you will come out of the road. That is nothing but respond. It means stimulus means changes in the environment, and respond means so acting to the stimulus is known as response. So for example, if you beat someone, okay, so that is a stimulus, and how the person is going to react, okay, that is known as response. Okay, that is the thing. If ant bites you, that is a stimulus, and how you react to the ant bite, that is a response. Okay, so you can go, if you think like this, you will go on and you will get a lot of examples in case of stimulus and response. Okay, so how the stimulus is carried? If ant bites you, you can identify the ant bite. Okay, so how you are able to feel the pain of the ant means the neurons. The neurons or nerve cells are going to carry such kind of information to the brain, okay, to the spinal cord and brain. Okay, it means we can understand that these nerve cells are nothing but they are involved in the carrying of information from the body part to brain, or you can call it from the surrounding to the brain. Okay, so here these neurons have they do not have regenerative power, means they cannot, if one, for example, if the nerve cells are damaged, the new nerve cells cannot be produced. So this is the reason they are non-regenerative, or you can call it as they do not have any regenerative power. Okay, so and there is no two neurons in the nerve cells have the same appearance. For example, it is like Aadhaar card. So in the entire India, your Aadhaar card number is unique. Okay, and your face structure is unique. No other people will have your face. Okay, in the same way, the nerve cells also the same. So all nerve cells will not look like. Okay, each and every nerve cell is unique, and one nerve cell will not resemble other nerve cell. Okay, so these nerve cells will be passed through neurons in the form of electric impulses. So these are some kind of signals. So from these signals, one neuron is going to pump the information in the form of electrical impulse to the other neuron. So, and also the brain and also spinal cord. So the neuron has three parts. They are, first one is the cell body. You can call the cell body as a cyton. Next is the axon. Next is the dendrite. So here you can see the head-like structure in the nerve. The head-like structure is known as cell body. It is also known as axon. You can see small branches on the head. Okay, on the branches on the head you will see some kind of branches. They are known as dendrites. Okay, so they are known as dendrites. And in the head-like structure that is known as cyton, it is going to have the nucleus. Okay, so cell body or cyton both are same. The cell body has different structures. They are cytoplasm. It is going to have the cytoplasm. It is going to have the nucleus. And Nissl granules are present in it. Nissl granules are nothing but small grain-like structures. They are known as Nissl granules. And also they have the dendrites. The dendrites are nothing but a branch-like structure on the cyton is known as dendrites. Here you can see the dendrites are the projections. So projection means something that is coming out, that is known as projection. Okay. And next is they are arising from the cell body, and they are sharp, branched, and more in number. Okay. This is about dendrites. Okay, so next one will be the dendrites are going to form web-like structure. Okay, web-like structure, and also they will show the projections arising from the cyton. Okay, so next even will be the axon. What is the axon? This is the longest portion or longest projection. Can you see a tail-like structure coming from the head? So this tail-like structure is nothing but axon. It is the longest projection when compared to the other dendrites. Okay. So this is the axon, and this axon is covered with fatty layer. You can see the brown color layer. So this is nothing but fatty layer that is known as myelin sheath. Okay, They are known as myelinated sheets, and in between myelinated sheets you can see there's interruptions or small gaps are present. So these small gaps are present in between the myelin sheets. It is known as nodes of Ranvier. Okay, so this is about the structure of nerve cell, and I hope this lecture is helpful to you. And if you have any doubts or questions, feel free to post in the comment section so that we can discuss your doubts or questions. Okay, and I hope this lecture is helpful to you.